Okay, it's 10 o'clock now. Good morning, everyone. Welcome to this month a CPA Member Trafficking Interest Group webinar. So today we're going to have our colleague Xiaolei Su, who presented last month as the host, and he will be introducing today's seminar speaker. Xiaolei.
Okay, yeah, thanks, Yihong, for organizing such a wonderful seminar series. It is my great pleasure to introduce my long-term friend and colleague, Dr. Pilong Li, who is currently a faculty at the School of Life Science at Tsinghua University. Pilong, I would say, is a pioneer and a world leader in biomolecular condensation, the fundamental principle governing almost all biological processes, including membrane trafficking. In my point of view, there are two seminal pieces of work that jumpstart the condensation field. One is a science paper by Tony Hyman and Cliff Brownring, which showed the liquid properties of P granules first in C. elegans. And the other one is a Nature paper published by Pilong when he was a PhD student in Mike Rosen's lab in UT Southwestern. In that paper, Pilong demonstrated the mechanism and the function of the biomolecular condensates. And he showed that the condensates were driven by weak multivalent protein-protein interactions, a fundamental mechanism that holds true for, I think, a lot of the following identified phase-separated structures. I think the committee in Stockholm will definitely have interest in this work in the near future. In 2015, Pilong started his own lab at Tsinghua University. He has been super productive and collaborative and published more than 70 papers since being independent. He served as a super engine to drive the growth of the phase separation community in Asia Pacific. And his research covered a wide range, including chromatin organization, transcription, regulation, autophagy, signal transduction, as well as method development to probe and engineer condensation. Because I'm interested, my personal research interest is on understanding how phase separation regulates membrane receptors. And one of my favorite work from Pilong is he engineered a multivalent ligand to drive the phase separation and control the DAS receptor signaling, which is truly innovative. I think today, Pilong is going to tell us about his recent work in harnessing condensation to clear difficult proteins. So without further ado, Pilong, the floor is yours.
Wow, after your introduction, I don't know what should I present, but of course, I highly grateful that Yihong invited me over for this discussion. Indeed, today I'm going to discuss with you actually it's a topic from the very beginning of my career till recently. I want to discuss a concept that is not being tested in the field yet. I think in our cells, there are plenty of degradation condensate or condensate function for degradation. So hopefully I can convince you. If I can convince you this concept, I will also want to convince you that probably we can harness this this phenomena or this concept to clear something that we want to clear, especially these pathogenic proteins. So let me start here. I think eukaryotic cells are highly compartmentalized. We all were familiar with what we're drawing in this cartoon. There are so many warm-colored membrane-bound organelles. But in here, we also deliberately draw these cold-colored structures. Those we meant to those non-membrane-bound organelles. These two classes of organelles to highly compartmentalize the cells. But I want to go one step further to let us think almost any biochemical activity in our cells is compartmentalized because the macromolecules are nanometer scale and the cells are tens of microns. So for other magnitude differences, if two macromolecules want to meet with each other, it's literally impossible if they do by random. So if you compartmentalize them, they can meet with each other, do something together. So that's my take with it. So almost most important biochemical activities in our cells are compartmentalized. Otherwise, it wouldn't be possible even. I'm from the phase separation field, as Xiaoli did very good introduction. Let me just jump over the two papers that he mentioned, Cliff Bryant's science paper and my own paper on the multivalency-driven LLPS. So we essentially at the time when we demonstrate that multivalent interaction can drive this physiochemical property phenomenon called liquid phase separation, we thought this phenomena actually probably responsible for the assembly of our cell membrane organelles. This hypothesis have been proven to be true over and over again. But over the last 12 years or so, people in the field, including Xiaoli, including many others, to show biomedical condensates have different kinds of functions in our cells. Anything you can think of that when you use condensation have advantages. So cells has plenty of examples like that. You can essentially inactive reaction or active reaction just by preferentially exclusion or colocalization of enzyme substrate, etc. But what is not being summarized here is how about the protein quality control pathways? Are those among those pathways is biomedical condensates or LLPS play any role there? So essentially, we know two major protein quality control pathways that we are familiar with, but including ubiquitin proteasome pathway and also autophagy pathway. I think many audience are expert here. So these two pathways are responsible for the majority of the protein damaged protein degradation, if you will. So we wonder whether LPS play a role in here. If it does, I really want to call those condensates as degradation condensates because, well, those two pathways are meant to be degrade the damaged proteins, organelles, etc. So this idea really stem out of a very simple experiment we did a long time ago. So we can culture a mammalian cell, then do a cell extract, those thick, transparent extract. We purify GLP ubiquitin or GLP-3 ubiquitin role or GLP-6 ubiquitin role, then mix the clear transparent cell extract with 600 micromolar GLP ubiquitin or 200 micromolar GLP-3 ubiquitin role or 100 micromolar GLP-6 ubiquitins. And only because 600, 200 times 3 and 100 times 6, they all 600. But only this higher valency ones can trigger a lot of condensate, as you can see here. So in my opinion, in our cytosol, there are plenty of protein factors. They are waiting for the ubiquitin chain to form, to be long enough, concentrate enough, then form a condensate for subsequent activity. We can guess maybe involving autophagy, maybe involve proteasome pathway, maybe involve signaling, etc. Indeed, when we do spin down and mass spec analysis, we found one molecule that many of us are familiar with, which is called PCC2 or sequencosome. This is well-studied selective autophagy receptors. We also find other players that I'll talk about later. So once we noticed this in collaboration with my colleague on campus, Professor Yu Li, we found out that actually he knows the PCC2 after starvation, it forms PCC2 body in our cells. This body, if you knock down downstream signals, don't allow the membrane to engulf those PCC2 body under EM, it's membraneless at this moment. So if actually you look at those PCC bodies, they sort of spherical, and two of them meet with each other, they can even merge together, suggesting this is a memberless liquid-like condensate in the cell. So to show what kind of signals to drive the formation of PCC body in cells, we use what we are good at, in vitro reconstitution. From literature, we find a protocol. You can purify ubiquitin monomer, purify AU1, purify this E2 complex, mix the three together. In the presence of ATP, it forms polyubiquitin chain. Without ATP, of course, everything is monomeric form. So if we don't have ATP, everybody is in monomeric form. You mix that with entire PCC2, 4 protein together, it's uniform distribution, nothing happens. But if you have ATP here, there are plenty of K63 ubiquitin chain form. Then this chain and PC2 together, extensive condensate formation, really tells us that it is the polyubiquitin chain and PC2 together drive the condensation in test tubes, likely also in the cells. Of course, the reason they can do that is because P62 is an oligomeric protein. This domain, PB1, is oligomer. Once you form oligomer, the UBA domain will become multivalent because in the same complex, multiple copies of UBA formed. Once you have a UBA, multivalent UBA domain, when it meet with polyubiquitin chain, each is identical subunit, then multivalent interaction, as what we showed previously, has a potential to undergo liquid-liquid phase separation, as shown on the right. Here we demonstrate we can use a linear chain. We can also use KC chain. And these two kinds of protein together form condensate. They are pretty dynamic. And also this in vitro reconstituted condensate is sort of functional because at least it can recognize LC3, which is the signal to be guided to form like autophagosome. So with plenty of other cellular studies, we propose that oligomeric PCC2, because it multimerizes UBA domains, once in the cells, you have enough long polyubiquitin chain. Those two multivalent entities will self-assembly via liquid-liquid phase separation. By doing so, it will automatically to assemble all those proteins that are meant to be degraded by autophagosome. So this is a sort of elegant mechanism because in our cells, the thousands kinds of proteins to be degraded by like autophagosome. The only thing in common is the polyubiquitin chain, nothing else. So therefore, using this chain as a signal, using self-assembly to form the condensate to automatically collect cargo is, I think, is pretty interesting or elegant mechanism. Let me see. Okay. Now, once I saw that, I think there's an analogy in our, for example, in any cities, we think we produce garbage every day. We see the garbage truck every day to pick up the garbage, but we rarely see where the garbage is being really processed. So, but in the cell, I think it's the same thing. You cannot do the garbage processing wherever you want. So if by this analogy, we think in our cells, there should be also garbage truck and the road to send the garbage to where the processing plant is. So now we ask the question, where do PC2 body forms, i.e. in the cell, where the garbage processing plant are? To ask this question, we collaborate with Bina, who is from Xinjiang Medical Institute. We pharmacologically perturb our roles in our cells. That's including active cytoskeleton, including microtubules. It turns out if we inhibit active polymerization or inhibit the L2-3 complex, which is a branch-acting polymerizer, those two pharmacoperturbation significantly inhibit P62 body formation, as you can see here, suggesting actin seems required for P62 body formation. Actin subsequently probably is the road towards a garbage processing plant. More than that, actually, when we do immunofluorescence staining of actin or up to the complex, which is colocalized with a PC2 body. So they are not required for, actually they physically localized with the PC2 body. And more, I think if you consider actin is the road towards garbage processing plant, so where is the truck that carry the garbage? So to do that, we just use a colocalization experiment to see the actin or branch actin specific myosins, many of those. Turns out myosin 1D is 100% colocalized with PC2 body at endogenous level or ectopic express level. They are 100% colocalized. Actually, it's more than that. If you knock down or knock out myosin 1D, it severely compromises PC2 body formation, suggesting myosin 1D not only colocalized with PC2 body, but also required for PC2 body formation in our cells. So not only cells, but also in test tubes. In the test tubes, only if we have actin, we have the machineries to produce branch actin, including up-to-3 complex and other subunits, as well as myosin 1D. Then you form effectively PC2 body. On the EMA to look at this. So with this, we demonstrate that in our cells, essentially, even PC2 body formation is somewhat compartmentalized towards the branch actin. The actin serve as a road. Myosin 1D at least serve as the truck to carry those small complexes to where branch actin are enriched. Then locally, both players, PC2 and ubiquitinate protein, reach above critical concentration, so undergo liquid-liquid separation from PC2 body. So using these two examples show us in autophagy how phase separation helps to assemble the cargoes and where it form this cargo complex for autophagosome formation. As I told you that during our previous exercise, we also saw the complexes from VCP. Let me see my timing. Sure. So VCP is, I think many of us know very well that is whenever you have a polyubiquitin chain involved process, you can find VCP. This is a central hub for many cellular processes where ubiquitin is involved. And the VCP is not function alone. It almost always requires cofactors. The most well-studied is the one NPL4 and UFD1. So in our mass spec analysis, we saw NPL4. So we wonder in proteasome pathways whether VCP is playing central roles in unfold the proteins to be degraded for proteasome degradation. So then we wonder whether the VCP somehow also form a condensate to perform its functions such as proteasome degradation. So as we know, P62 form a PC2 body upon starvation. So we also create a cell that endogenous knocking a GLP to VCP. And upon starvation within hours, it form extensive VCP positive condensates. So let me just quickly show this condensate colocalized not surprisingly with proteasome. It also colocalized not surprisingly with K48 polyubiquitin chain. So with plenty of biochemical chemistry, we think that PCC2, VCP, and polyubiquitin chain together, just like PCC2 and polyubiquitin chain, they can form condensate upon starvation and other stresses. Upon doing so, it will also enrich proteasome, maybe other E1, E2, E3s to locally perform protein degradation like a function. So we think this is also a kind of degradation condensates. Well, this work was a collaboration with Professor Pan Man from Shanghai Jiaotong and Liu Li from Tsinghua University. I failed to find their photos here. So those two PC2 body and VCP body are the examples that we are studying in my lab. Actually, there are plenty of other examples in the literature. For example, early 2000, Yosuda and colleagues show that at osmotic treatment of cell, they form very beautiful nuclear condensates those endogenous ones, I should say, those condensates form at least in part with a divalent UBA domain from RAD23B and also polyubiquitin chain. So those multivalent actions, in part, responsible for this condensates in which enrich with proteasome, probably also VCP. And this condensate is responsible for degrade some substrate. What they discover is a subunit from a large ribosome. I think there should be plenty more other condensates, substrate for these condensates. There also ubiquitin-2 is a proteasome shuttling factor. It's a colleague in the field show that it feels separate for its functionality. A different polyubiquitin chain linkage have a differential regulation toward its functionality. And also here's an interesting one. SPOP is a E3 ligase subunit that is a polymer, right? And it recognizes many oncoproteins, for example, DASX. So in the cell, if DASX as an oncoprotein are expressed, then the two together reach the critical concentration. They form nuclear condensates in which, of course, you reach all kinds of ED1, E2, E3s to ubiquitin DASX for degradation to keep those oncoprotein in check. Any mutations abolishes oligomerization or abolishes binding to this oncoproteins will lead to cancers. This is a more interesting example. So the evolution, in order to keep in check of the beta-catenin-like important transcription factors, it evolved a phase-separate condensate, previously called destruction complexes. So my colleague Chen Yeguang demonstrated that this degradation complex actually is a phase-separate entity that just to evolve to keep the beta-catenin and other important molecules from getting to nucleus for aberrant transcription until when pathway is turned on. There are more examples I'm not going to enumerate. Essentially, what I told you is that there are plenty of different kinds of condensates in our cells that are evolved functional in degrade, I think, substrate that can be through proteasome, that also can be through autophagy. Now we talk about, now let me just move on to the second part of my talk. If I convince you to a certain degree that there are plenty of degradation condensates in our cells, so what can we use it for? Essentially, we all know that the cell has in a homeostasis status, so protein production, degradation should be balanced. If it's in balance, we'll get in trouble. So recognize this trouble in academia and industry. People invented targeted protein degradation in the last 2 decades, if not longer. For example, people invent a PROTECT or molecular degrader as a small molecule that trying to degrade the unwanted proteins. There are also new neural development just hijacking lysosome pathways, also degrade intracellular or extracellular or member proteins. Again, trying to restore the protein homeostasis, hopefully to cure diseases, if you will. So to us, the conventional targeted protein degradation, for example, PROTECT or molecular degrader, is essentially a one-to-one strategy. Somehow you like bridging a protein of interest to a E3, for example, and hope that E3 will polyubiquitinate the protein of interest, then proteasome will kick in to degrade the protein of interest. Now, with the realization that in the cell, there are plenty of condensates that are help degrade, can we just deliver the protein of interest or RNA of interest into the degradation condensate to see whether the cell will also take care of it automatically? To do that, we want using like the well-studied degradation condensate. For example, in the nucleus, it's a PC2 body, as I just showed you. It so turns out that PC2 in nucleus, it can also form phase separation with the polyubiquitin chain. And then in the nucleus, it will harness proteasome for degradation of its substrate. Essentially, PC2 is a magic molecule responsible for degrade anywhere you want. So based on this knowledge, we think maybe we can invent a reagent that bridge the protein of interest and the p62, so that the bridging will bring any protein of interest into p62 body in the nucleus or in the cytoplasm, if you will. So we give it a name called p62 body, p62 degradation factory based on bispecific nanobody. In short, PDF bin. Bin is like a trash can. But what we think is we are not good at chemistry. We don't want we cannot produce PROTAC or molecular glue degrader at will. What we can do is we can use learn from evolution. Evolution was able to produce nanobody as a domain upon from like a llama, camel, ray, and a shark. So they have a special IgG that is only heavy chain. So the variable region is a single domain. You get it. I think you can reach this kind of antibody against any antigen if you want. So we, with our collaborator, we reached quite a number of nanobodies against a human PC2 body, PC2 protein. So it's suffice to say that we have quite a few. For example, D9A, E12C, and A1E. They bind to different part of the PC2 protein. So once we have those nanobodies, we think, okay, maybe we can do proof of concept type experiment. For example, from literature, we know that there's a very good GFP nanobodies. So we fuse the gene encoding PC2 nanobody and the GFP nanobody. So this gene expression in our cell, then it become a bispecific nanobody. And on one end, get bind PC2. On the other end, combine GFP and whatever GFP fused protein you want. So now we have a cell that stably express GFP, into which we introduce the fusion by specific antibody. All three of them efficiently decrease the GFP signal in the cell, suggestive degradation. Now we have this tool in mind. Then we test all kinds of GFP fused target proteins. Essentially, we pick one of the nanobody bind to PC2 specifically, and then fuse that with the gene, which is a nanobody bind GFP. So we establish a stable cell line that can induce expression of it use doxycycline. So we have M-cherry just to show whether it's expressed or not. So in this stable cell line, we introduce all kinds of GFP type protein of interest just to see whether they can be linked together with the PC2, whether can induce its degradation. Using this cell line, introduce with like GFP labeled TD43 as a nuclear protein, GFP G3BP1 as a cytoplasmic protein, GFP tetraspanin as a membrane tether protein, as well as a Huntington polyQ as a aggregation protein. So let's just use this as example. Of course, if you don't induce it with doxycycline, just treat it with the DMSO, the bispecific nanobody is not being expressed. So TDP43 and P62 are not talk to each other whatsoever. But once you use doxycycline, induce the expression of the bispecific nanobody here using the red color. This nanobody will bridge TDP43 with P62 to form P62 body here. So the same thing applies to all the other proteometries that we tested. We then use a Western blotting to show that upon expression of bispecific nanobody, they all introduce efficient degradation. So the more yellowish, the more degradation you can see from Western. And this degradation is PC2 dependent. It's another name called sequestrome. If you knock out PC2, the degradation is gone. So essentially, our PDFB can efficiently degrade all kinds of GFP type protein of interest. During this process, we noticed something interesting. As we know, TD43 is a key pathological, at least a biomarker for all kinds of neurodegenerative diseases, including ALS, including AD, for example. So we also know that TDP-43 can self-associate to phase separate or form aggregates. So during our bridging experiment by bridge TDP-43 with P62, as I told you that P62 is oligomer, so that will increase the valency for TDP-43. That exercise alone induce massive amount of P62 body, as you can see here. So essentially, you can so if you induce expression of bispecific antibody to bridge TDP-43 and PC2, induce a lot of PC2 bodies showing here. And sure enough, you form a PC2 body, the TDP-43 is efficiently in time-dependent manner and dose-dependent manner being degraded, presumably via PC2 body formation. So of course, we also pick up endogenous targets to show that we can essentially target degradation of endogenous ones, including STAT3. So this is an oncoprotein. So we established a cell line that inducedly express bispecific antibodies. On the one end, it can bind PCS2. On the other end, bind to STAT3, endogenous STAT3. This is a monobody from literature that can bind STAT3 specifically and efficiently. Once we do that, once we express the bispecific antibody, now the color is changed. Here's green is for bispecific antibody. That bridge STAT3, endogenous STAT3 with endogenous PCS2, we form again extensive PCS body. Once you do that, the STAT3 is being degraded in a time-dependent manner or dose-dependent manner. And it turns out those degradation is lysosome dependent because use bifidobacterium inhibition, then the degradation is gone. So more importantly, because it's oncoprotein or the driver for some non-small cell lung cancer for this A549. So in this cell line, upon degradation of STAT3, in terms of collagen formation assay or wound healing assay, show that the degradation of STAT3 is compromise this cancer cell line such kind of cancerous activity. So with that, I want just to summarize, at least in terms of PCC2 degradation factory, that including PCC2 body in cytosol and also PCC2 condensate in the nucleus. We can harness this kind of degradation condensate by recruiting the protein of interest or RNA of interest, if you can, into this condensate, and the cell automatically help us to degrade that. Those are some proof of concept type experiment, but we really want to apply this idea to other diseases like cancer, like chronic rare disease, especially neurodegenerative diseases. So for those kind of efforts, we just have to work in progress. We don't have like a solid story to share with you guys. Hopefully we will come up with it soon. So with this, I would like to just draw the conclusion that all the experiments have been carried out by current or previous trainees of the lab. Some reagent appeared the bill, and I only responsible for discussing with you guys. Thank you.
Okay, great. Thank you so much, Pilong, for this, I think, truly innovative work by harnessing the degrading condensates to clear actually a variety of cellular proteins, including these pathogenic proteins. I think this technology will have a broad use for not only understanding biology, but for a lot of potential therapeutic use. So now the floor is open for questions. And if you have any questions, please feel free to type in your question in the chat box, and I will read it one by one. But also, if you want to directly ask your questions, just raise your hand. Okay, so I think the first question comes from Mao Yuxing. So how does MYO1D recognize nano p62 bodies?
Oh, that's a good question. We have biochemical colocalization and the IP data to show that the cargo binding site of myosin 1D is able to bind to PC2, I think. It's in the paper, we have extensive characterization, so I just don't have time to discuss here for the sake of time.
Okay, great. So next question actually come from Yihong. So Yihong, do you want to directly ask the question?
Yeah, thank you, Pilong. This is really inspiring. So yeah, from myself, it's also very interesting in protein quality controls. I'm very cool to see this new ideas, and I wonder, what is the evidence that this condensations can actually active degrading proteins as opposed to just being some kind of a storage? I mean, in the protein aggregation field is always being debating as to, for example, the aggresomes or protein aggregates, whether they're actually helping cells or they're actually toxic. And when you see these condensations and you do have machineries there, ubiquitin, etc. So can you actually disrupt these condensations and then show it's actually affecting either autophagy or proteasome degradation?
Oh, actually, we see the target is gone. Is that evidence to be degraded?
You mean if you, for example, if you knock down MAO1D, presumably would not form this P62 body,
Yeah.
right? On starvations.
Yeah.
Does it affect P62 mediated autophagy or degradation?
At least the ubiquitin signal is greatly enhanced. Again, it's in the paper. I just didn't have time to show it. So if you knock it down, knock out myosin 1D, or you pharmacological perturb all those trucks, myosin trucks and roads, then the degradation, a lot more PC2 body formation. No, a lot of ubiquitin proteins on the gel, I should say.
This is detected by ubiquitin staining, you say, right?
Yes.
Okay, I see. And I have another question about the VCP body. So apparently VCP has a lot of functions, both degradation and non-degradation functions. And one of its key functions is to degrade photoproteins at the endoplasmic reticulum. And this works together with, membrane-associated E3 ligase. And I wonder, in your VCP, when you form these VCP bodies, do you see those ER-associated ubiquitin ligase in there as well?
Yeah.
In other words, whether this is something dedicated for cytosolic proteins or actually can also use to degrade membrane material, membrane associated.
I think VCP condensate is dynamically involving all kinds of degradation. So for example, if we're using the ER stress, we saw a different morphological condensates. In wild type, it's a ring-like structure. In the ring, it colocalizes very beautifully with PC2, LC3, HERD1, CL1. Anything you think involving degradation is there and beautifully colocalized.
Cool.
And all those are endogenous, not overexpression.
Okay. That's great. I see somebody raise hands in the audience.
Yeah, so Ethan Sanowski. So Ethan, are you there?
Yes, great proof of concept.
Thank
I
you.
think I heard you say, as you began displaying the proof of concept, that you couldn't identify a small molecule that would bind P62. And I just wanted to follow up on that to understand, is that just something that hasn't been done yet because there's not a published P62 ligand that you could use to create something more similar to published small molecule PROTACs? Or is there some intrinsic difficulty in developing that? And is that sort of a current direction to figure out how to make this into something that could penetrate a cell membrane and be more easily used in a therapeutic fashion?
That's a good question. Actually, when I started work on PC2 body, I immediately initiated the program to use DEL to find small molecule that combine PC2. But because PC2 is so modular, we failed. We tried extensively, spent a lot of money. We didn't find a effective binder, so we give up. Later, somebody in the literature reported some small molecules that they claim to bind PC2. We weren't able to demonstrate effectively. So I think it's very it's fair to say that it's very difficult to find small molecules that can bind PC2 specifically with high affinity. I think that's a fair statement. Otherwise, we would be allowed to have a chemical-based protect or molecular glue degrader.
Do you have a strategy that you're planning in terms of how to actually get a potential therapeutic based on this mechanism into the cell, or are you currently in concept limited to perhaps being able to deliver something like this via gene therapy to a relatively small number of cells?
Yeah, you mean the PDFB idea, right? So we are actually to apply this to ALS and AD like PD targets to see at least at animal level whether it have efficacy. If we do, we'll be pretty happy already. But so we are trying. Thanks.
Okay, great. Thanks, Ethan. Next question comes from Hu Fenghua. Amazing talk. Will this method eventually lead to depletion of P62 or affect the function of P62?
The reason I'm in for this pathway in comparison with other protect-like structure, like a pathway, I feel that the capacity of PC2 is enormous. Evolution picked this molecule, just do as little as possible, as much as possible. They can even infection, mitochondria, all kinds of damaged stuff will be degraded in this pathway via PC2. So we are not that worried about the depletion of this pathway.
Yeah, I guess a related question will be, do you see any perturbation of the regular P62 dependent degradation when you have this inducible degradation?
Actually, PC2 function when it's being starved or something. Normally, even you knock out PC2, the cell is happy. So I don't know what kind of activities depend on PC2 to test around.
So for example, in the starvation, when they are supposed to degrade, let's say, a lot of intracellular component, and on top of that, you have your induced this like bispecific nanobody. So it's basically whether this inducible this degradation will interfere with the physiological degradation pathway.
Yeah,
I guess that's the question.
Yeah, that's a good question. I was hoping that the bispecific nanobody just take the free ride.
Okay. But I think it depends on the abundance of P62 condensates in the cells, right? So if it can provide sufficient amount, then probably it wouldn't affect too much.
Yeah, this pathway, I feel maybe a more expert in the audience. I feel it's very like versatile. It can be induced and it can be regulated by phosphorylation, etc. Well,
okay, that's
a
great.
hope. That's just a hope.
Yeah, that's great. So next question coming from Liu Yanfeng. Professor Li, great talk. I have a general question about protein activity in phase separation. Why are some proteins sequestered? Why are some proteins sequestered are deactivated, while others become activated?
Yeah, it's all depends on what kind of proteins and what pathways. Just give you two examples. One is called processing body, the other called stress granule. So if you recruit mRNA into stress granule, it's a kind of deactivation from degradation from translation. But if you recruit to processing body, it's activation for degradation of mRNA, decapping, polyubiquitination, poly-A tail, then degrade the whole mRNA. I feel this is all case by case. We cannot just generalize to have a generalized answer to all of it.
Yes, yeah, I think it's all like context dependent and protein dependent, right? Okay, that's great. So Yuxing, I think you have another question.
Unmute.
Oh, sorry. Yeah. Very nice talk, Pilong, and I enjoyed it a lot. But I have a question about regarding your PDF Bing technique.
Sure.
In the experiment, when you, I wonder when you compare the protein level for point of interest for degradation, did you use the whole like cytosol, or it's just soluble fraction? Because I feel like somehow some protein maybe you see if you use the whole only cytosol protein, some of the protein may be sequestered and forming aggregates, and that's why you see a low level of the protein you want to degrade. So I just want technically I want to clarify is what kind of sample you use to measure the protein degradation.
Yeah. I think we just use the whole cell because for SDS page, the buffer is strong enough. We didn't take the effort to just separate cytosol from like a nuclear, separate the pellet from a supernatant. I think we just do the lazy job at this moment. So the whole thing is being collected and loaded onto the gel. So I feel it's the total. It's not. And for those the ones that we are tested, there's no like sequestration and precipitation that is not being resolved in the gel. I hope I answered the question.
Yeah, yes, great, thanks.
Okay, great. So Pilong, I also have a few questions if you don't mind. So what are the degradation efficiency, or what do you think is limiting the degradation efficiency? Let's say if we want to achieve a complete depletion of the target protein, is there anything that we can do? Because based on the Western, I saw there's still some left. Is that because of the protein, the target protein is still constantly being produced? Is that like what is the limiting factors to control the degradation efficiency?
Certainly the continuous production is one thing contribute to the residual level, but it's more than that though. Some proteins are degraded easier than others. Let me just show you that a panel, although it's extra no, where is it? You can see that like GFP is less degraded. TDP-43 is more. Tetracycline is more. So the main proteins are sort of less. Where's polyQ? Q is sort of less. It's what I see is if we have interest disorder proteins, that tends to be degrade more efficient than the others. We thought that at least interest disorder is a better substrate for proteasome, and also it will enhance the PC40 formation.
I see. So when you're trying to degrade the membrane protein, did you see let's say it caused any membrane defects or compromising the membrane integrity?
We haven't do very careful analysis. We just did imaging to show their colocalization. This is tetracycline. And also use Western to show PD-L1, APP, and tetracycline is indeed degraded. We haven't looked more carefully what happened to the membrane, etc.
I see. Did you see the P62? These condensates are mostly in these membrane protein case. Did you see they are mostly in the intracellular case, or actually they can also go to the plasma membrane? For example, PD-1 is definitely on the plasma membrane, but are they predominantly actually in the endomembrane for the P62 terminal?
For the visible ones, I feel that already internalized.
Okay, I see.
Yeah, we haven't looked careful enough. This is just a proof of concept that it can degrade.
I think probably it's related to the size or to the curvature of the membrane that probably also had the influence of the interaction between the P62 and these things. My maybe another question is, have you tried to use this to, let's say, degrade any let's say organelles or vesicles?
That's an interesting question. We didn't do anything about that, but during a TPD, Keystone conference, I was presenting my poster. I saw an almost identical poster from Novartis. Let's use target mitochondria, 2p62 body, identical design. So yeah, Novartis did this for us, I should say. Yeah.
I see. Okay, that's great. So actually, I saw a WG. We just have a question.
Maybe.
Are you there?
This is Wei. Hi,
Wei. Oh, sorry.
Pilong, it's a wonderful talk.
Thank you.
I think Xiaolei already asked the question I intended to ask. By the way, it's a great talk. Thanks for the seminal work to the field. It's great.
Thank you. Yeah.
No, my question is related to Xiaolei's question, like organelles and also the PM tetraspanning and for example PDL1, you are targeted to the PM because there's such a physical, membrane insertion, a large entity. How can that be degraded? And how, I mean, we show some degradation by Western blotting.
Yeah, that's
I
all we
missed this, but how can they achieve this? Since they're so big. So they're in the membrane, for example, especially in the plasma membrane.
To be honest, we didn't study the kinetics in the detail how pieces to be bridged to those entities, then form autophagon. We haven't do any of those. We are just learn. I take the advantages of endogenous pathways. Just hope they works, and they do. Yeah.
Cool.
So, I have a technical questions. I know there's another question in the chat box. Maybe I can squeeze in. I just wondering, do you know the epitopes where your P62 nanobody target? The targeting the ubiquitin binding domains or the substrate binding domains? Or?
We have three. A1E is the bind ubiquitin UBA. This E12C is here and D9A is here. Now we have a lot more binding.
Which one works the best? Do you know?
All three works. There's only one didn't work. I haven't shown you that. I don't remember. Don't know why it didn't work. The binds elsewhere. Probably perturb the function of P62.
I see.
We have the fourth one didn't work, so I only show the three that works.
Thank you.
Okay, so I think there's one more question from Beijing in the chat box. How do you deliver your PDF beam into the live cell? Any cell preference? And how do you control the optimal amount? Yeah, I think probably this question has been covered before.
Actually, this is a good question. The short answer, the multiple questions. Control the expression level is something we should follow up to make it more smart design. At this moment, we didn't. Just use like a generic promoter. So then the related for delivery at this moment, we only target using AAV delivery because there's other ways it's hard, not efficient. Therefore, the disease we're thinking about all those post-mitotic ones like neurodegenerative diseases.
Okay, great.
Thank you.
Okay, if there are no more questions, let's thank Pilong again. And this is a really, truly, I would say, innovative work, which I think will be broadly applicable to both integrating and for understanding a lot of biological processes. Thank you so much, Pilong.
Thank you, Xiaowei.
Thank you.
Thank you, Yihong. Okay.
Thank you. Bye bye, everyone.
See you guys around.
Yeah.